So, hello everyone. My name is Tanu. I'm your TA for this circular dichroism and Mossbauer spectroscopy course. So, here in these interactive sessions, we will be solving some doubts related to the previous week content, and mostly we try to take some sample questions so that based by solving those questions and by understanding the pathway to solve those questions, you will be able to solve the main assignment questions. And if you have any doubt with any course content, maybe like you have doubt in some topic, or you want to like clarify any doubt, you can ask in these interactive sessions. So here I would like to ask your feedback if you are if you are able to understand the questions or the content being discussed in these interactive sessions. So I am requesting you again. So if you have any doubts, if you have any feedback or any suggestions, please reach out to me. I'll share my email ID. Uh, just a minute. I'm writing it over here, but from the next session, I'll make sure I add it into the slide only. You can mail me on this email ID, and also we have discussion forum. You can directly post your doubts in that discussion forum also. And even if you are unable to ask a question through that, you can share your problems on this email address. I hope the content, like how to find out the symmetry group and the symmetry elements of a particular molecule, is clear for you by now. And we have taken an ample amount of example till now, and that must be that and you must be now well equipped with the knowledge how to find out the different symmetry elements in a molecule, and how these collection of symmetry elements form a particular point group to which a molecule is associated to. Here, based on these symmetry groups, we can directly find out whether molecule is chiral or not. In this week 3 lectures, more emphasis has been given on chirality and the application of and the importance of chirality in the biology, like in our biological systems, plants, animals, whatever, enzymes, proteins, carbohydrates. So in these kind of things, how chirality plays an important role. So this has been taught very well in the main lectures, but here I will be just taking a few examples on chirality and some amino acids question, because these were the highlight of the this week 3 content. I hope it will be helpful for you. So, if based on a if based on the symmetry elements and point group of a molecule, if you if you have to find out whether the molecule is whether the molecule is chiral or not, you can find it out directly if a molecule possess possesses an improper excess of rotation This symmetry operation involves rotation around an axis and followed by a reflection through a plane which is perpendicular to the principal axis, which is perpendicular to rotational axis. If a molecule has this improper axis of rotation, then that molecule will be achiral. Will be achiral. So now you just have to. We have already discussed the flow chart to trace out the different symmetry elements and the particular point group of a molecule. So following that flow chart, you can find out the point group of a molecule, and then you can assign whether the molecule has an improper axis of rotation, and based on that you will see that only Cn and Dn, that is single axis rotation group, single axis rotation group Cn, and dihedral group Dn will be chiral, because these are the two point groups which do not exhibit improper axis of rotation. So based on these point groups you can directly find out whether molecule is chiral or achiral. An object is basically considered to be chiral when it is not superimposable posable on its mirror image. So based on this, we will take some example, like some basic molecules, some basic examples where we will find out whether a molecule is chiral or not. Some organic moieties we will be taking. Okay, first is the number of asymmetric carbon before that. Hello, Anita. Can you hear me?
Yes, ma'am.
So, are you following these interactive sessions?
Yes, ma'am. I'm watching.
So, are these helpful for you? And do you have any doubts or anything you particularly want me to discuss?
Mm,
Yes. Okay, it's okay. It's fine. If you have any doubts or any feedback or suggestions, you can write in the chat box also. Okay.
Okay, ma'am.
Yeah. So let's continue our question. In the below structure is this asymmetric means chiral. That is, there is no symmetry present. So, Anita, can you tell me how many number of chiral carbons are present in this structure? Because you are the only student, I want it to be a bit interactive. If you are okay with that, if you are comfortable.
Only one, ma'am.
Okay, right. So, which one is that?
The second carbon atom.
This one, right. Very, very
Yes,
good.
ma'am.
Yes. So, here we can see for a carbon, for a molecule with carbon atom which has four different substituents is said to be asymmetric or a asymmetric or chiral center. So here only this center, if you see, we have this one bromine atom, then the second substituent is this group, and the third substituent is this particular group, and the fourth is hydrogen. So this is the only carbon atom which has four different substituents on it. Otherwise this center has two hydrogen atoms which make it an achiral center. Okay, so this was
Ma'am,
very basic. Yeah.
ma'am, I have some doubts on point group. Can I ask, ma'am?
Yes, please.
For the molecule NHF2, how to assign point group, ma'am?
The molecule,
NHF2.
can you write in the chat box, please? I can't hear you properly. Please write in the chat box, I'll solve it. NHF2. Okay. So here this is similar to ammonia, NS3. Ammonia belongs to C3V point group. Okay. But here in this molecule, we have two different substituents. Like instead of, we don't have three similar substituents, that is hydrogen, but we have two fluorine atoms also. So if I represent this molecule like NHF, uh, sorry, I just give me a minute. I have this problem with this pen.
Okay, ma'am.
So here we have fluorine atoms. Now, if we like, I'm taking this, I'm representing it in a planar form now. although these atoms are not in planar form, this is pyramidal structure. So here, if you see, we don't have a C3 angle, C3 rotational axis in this molecule, because if we move this molecule at 180-degree angle, then we won't get an equivalent configuration. Instead, we have to find out whether we have a lower degree of axis of rotation in this molecule. So if I NHF, so if you see, if you rotate this molecule at 180-degree angle along this axis of rotation C2, which is passing through the NH bond, then you will get an equivalent configuration, right? That is, this will become F1 and F2. This is an equivalent configuration. So we have C2 axis of rotation first. Now we will have to find out whether it has a molecular plane, that the horizontal plane in this molecule. So perpendicular to this, if we draw a plane, then we won't get an equivalent configuration. Instead, we have a sigma v plane which is passing through this C2 axis of rotation. So we have this sigma v, and also we have the molecular plane of this molecule, which is the plane of the board. So we have two sigma v. So based on this, we get C2v point group in this molecule. Did you get my solution?
Yes, ma'am.
Is it clear? Please tell me honestly, because you can see here we don't have a C3 axis of rotation, because when we
No,
rotate the molecule at 120-degree angle, we won't get an equivalent configuration. The H and F atoms will replace each other. However, after C2 axis of rotation along this NH bond, we will get an equivalent configuration. Okay?
ma'am,
Yes.
ma'am. But in yesterday's exam in our college, the answer key is given as CS point group.
Which point group?
CS.
CS. That is, it only has
Identity
sigma
and
plane.
plane.
No, no.
Yes,
CS
ma'am.
is only sigma plane. That the
Yes,
molecule has
ma'am.
only sigma plane.
Mm.
Okay, just a minute.
Okay, ma'am.
Okay, so here, since I have done this mistake, that I have made it into a planar. When we are using it with three hydrogen atoms, we can represent this molecule as in this triconal planar way, but here it is not possible because we have two different substituents. Then we only have a sigma plane, that is this plane NH, which will bisect the two fluorine atoms opposite to each other. That is, it will reflect only these NF and NF bonds. So I did a mistake here. I'm sorry about that. I didn't notice it. Okay.
Okay, ma'am.
Yeah, so see these nitrogen atoms, and then we have fluorine atom, and then hydrogen atom. So through this NH bond, I am bisecting it, and then we are getting these NF bonds as reflection of each other. So that is the right answer actually. You are right. But do you get it how it is only sigma plane?
But why rotations are not possible here, ma'am?
See, the rotations which will be present in this molecule basically. Let me add the slide. This is NH. So here in this molecule, in ammonia molecule, it is similar to ammonia molecule only, na? Only the substituents has been changed, but the geometry is similar. Okay, if I represent it as fluorine atom, and then NH atom. So both have pyramidal geometry. When you rotate this molecule at C3, that is 120-degree angle, H1, S2, and S3, you will get H1, S2, and S3. This is the equivalent configuration you get on C3 axis of rotation. But here, if you do this C3 axis of rotation, what will you get? Now, see, here you can't differentiate between the hydrogen atoms, like which hydrogen atom is in the plane, which is on dash, and which is on wedge bond. But here you see the fluorine atoms, which were on dash and wedge bond. This is dash bond, and this is wedge bond, are now in plane. So this is not the equivalent configuration, right? Because hydrogen was in the plane now.
Okay, ma'am.
So here we don't have C3 axis of rotation. So the minimum axis of rotation then we can have is C2 axis of rotation, because C1 axis of rotation is 360-degree rotation, which is always present in any molecule, which is basically identity operator. Okay. Then only we can check C2 axis of rotation. So what are the possible ways that we like rotate the molecule so that we get an equivalent configuration? In this particular
Um,
geometry, we can't.
when axis is passed through nitrogen and hydrogen bond,
Yes.
we get C2 axis.
Sorry.
Ma'am, when C2 axis, we can pass C2 axis through the nitrogen and hydrogen bond, so that we will get equivalent structure.
That was what I supposed. Okay. So just a minute, I'll show you some tutorial. Okay, I'm sharing my screen with you where you can visualize it in 3D. Okay.
Okay, ma'am.
Can you see this molecule? This is ammonia molecule. Anita, can you see this?
Yes, ma'am.
Okay. Great. So here I'm showing you how this C3 axis of rotation is present in this molecule. This is C3 axis of rotation. Then we have only reflections sigma v planes in this molecule, which are passing through each NH bond. So here in this molecule we don't have with 3C2, like in a molecule we can only have NC2, that is 3C2. But here in C3 molecule, which is present in ammonia, C3 point group, we don't have any C2 axis of rotation. Do you get me?
Yes, ma'am.
The only symmetry elements present in this molecule are C3 axis of rotation and vertical planes. How can you if you pass the C2 axis of rotation through any NH bond, let's say.
Mm.
So it will So it doesn't have C2 axis of rotation in it based on this NH bond, because I can't visualize it that way. But I will try to clarify it in a better way. Okay, in the next session maybe.
Okay, ma'am.
Yeah. Okay, I'll look into it.
Ma'am,
Yes. Yes, Anita.
ma'am, I will clear it in our next session.
Okay, sure.
Okay, ma'am.
So we were discussing these examples on chirality in a molecule. Let's take another example. The total number of optical isomer in the given compound. So the formula for number of optical isomers is is 2 raised to n minus 1, where n is number of asymmetric or chiral centers. Okay, so now tell me how many chiral centers are there in this molecule?
Four,
What?
four, ma'am.
No. So here we see this is sp2 hybridized. So here its two bonds are already occupied by these oxygen atoms. So any molecule to be carbon atom to be asymmetric, it has to be have sp3 hybridization, because the concept is all four valencies are occupied by four different atoms. Okay, but here two of the bonds are being occupied by oxygen, so it can't be a chiral center. Similarly, this can't be a chiral center. So here, just a minute, I have an important call. हेलो। भैया मेरी ना क्लास चल रही है, मैं नहीं आ पाऊंगी। 8 बजे तक है भैया। तो तुम्हारे लिए क्या लाते बताओ भैया। कुछ नहीं भैया, मैं आप मुझे चाय पिला देना। अरे लेट जाएगा, तुमको तो पिला दूंगा, बट फिर भी बताओ क्या खाओगे तो नहीं नहीं भैया, मैंने मुझे सच में खाना कुछ नहीं है। क्या? 8 बजे क्या लैब आओगी? नहीं नहीं भैया, लैब तो नहीं आऊंगी। अरे ठीक है, तो हम हॉस्टल भिजवा देंगे तुम्हारे, बताओ क्या लाए। कुछ भी भैया, दे वो थिनलेस। हाँ भैया, मेरी क्लास चल रही है, मैं आपको फिर फोन करती हूँ, ठीक है? हाँ, थैंक यू। Yeah, sorry for the disturbance. So here we have symmetry in this molecule, and these are two chiral centers which are present in this molecule. So here, because you can see that this is one substituent, second is this hydroxide functional group, and then third is this moiety, and the fourth is hydrogen atom. So these are two stereo centers present in this molecule. So the number of optical isomers will be 2 raised to 2 minus 1, that is 4 minus 1 equal to 3. So here we have 3 optical isomers. So basically you have to find out the right number of chiral centers in a molecule. Okay.
Yes, ma'am.
So now I'm drawing this molecule. Help me find out the number of optical isomers for this molecule. COH H. So now tell me how many chiral centers are there in this molecule?
Two,
Right. So which one are these? I'm labeling them as 1, 2, 3, 4, and 5. So which one of these are
second and fourth.
second and fourth? Right.
Fourth.
Because this atom here we have, if we draw a plane passing through this molecule, then we will get a equivalent figure. So here this molecule is achiral because these substituents are same. So this is a chiral center, and these are Second and fourth are chiral, where these methyl groups are obviously achiral because these have three similar hydrogen atoms. So now let us move to these questions on chirality and amino acids. So the questions are visible on the screen. Uh, Let me ask you the answer for the first question. The chirality of an amino acid results from the fact that it's a carbon. It's carbon. What and the options are: has no net charge, is a carboxylic acid, is bonded to,
Option C.
yes, option C. We have already discussed when a molecule is said to be chiral. Whether it is an amino acid or any other molecule, we just have to find out the chiral center, and the carbon atom should have four different substituents on it. So the right answer is C. Now, out of the 20 standard amino acids, only which amino acid is not optically active, and the reason is, so are you aware of the 20 naturally occurring amino acids?
Yes, ma'am, but not clear about its structure.
Okay, so
It may be D or E. If we
okay. Okay, fine. So before that, let us discuss the different type of amino acids present in a molecule. Would you like to make a note of these amino acids, or will you be following the main lectures?
I will take note of it, ma'am.
Okay, okay. So let's discuss this then. So here we have 20 naturally occurring amino acids, and we know out of out of these there is only one amino acid which is achiral acid, which is achiral, and that is glycine. Why? Because an amino acid has two functional groups which will always be present in that is amino group and this carboxylic acid group. This amine functional group and this carboxylic acid, and it will have hydrogen atom, and there will be one substituent. Based on this, there are 20 different amino acids. Based on this are substituent, and it can be aliphatic, only hydrogen. If hydrogen atom is present, then it will be glycine, and if it is aliphatic, so I'm starting from the very beginning. Amino acids, as we discussed. So based on this basic structure, there will be 20 naturally occurring amino acids. This R group, these can be chiral and achiral. Only one amino acid is achiral, and 19 other amino acids are chiral. So if a molecule is chiral, then it will have enantiomers. So there are two enantiomers for amino acids, that is D enantiomer and L enantiomer. And how do you find out which is D and which is L? Just a minute. Hello. So we find out whether an amino acid is D enantiomer or it's L enantiomer based on the cone rule. This cone means CO is carboxylic acid, R is aliphatic, aromatic, polar, or any other functional group, and N is amine group. So firstly what you have to do is put hydrogen atom on the back, that is away from the observer, that is on the dash bond. Second, then orient in the following manner: where you assign COOH as the first priority, and then this R substituent as the second priority, and amine as the third priority. So if you have COH, R, NH2, and I have already put the hydrogen atom on the back. Now tell me which enantiomer is this? We will assign COH as the first priority, then R as the second priority, and NH2 as the third priority. And we will move from first to second, second to third. Okay. So this is anti-clockwise configuration. Anti-clockwise rotation, which is equivalent to L enantiomer. So if we are orienting in an anti-clockwise direction, then that molecule is the L enantiomer. Okay.
Okay, ma'am.
Similarly, now again, if we assign the priorities, make sure the condition is your hydrogen atom is always on the back, that is on the dash bond. So now if you orient through this, then you are moving from 1 to 2, 2 to 3, and then back to 1. So this is clockwise rotation. So clockwise rotation is equivalent to your d enantiomer. Okay. So if a molecule is a chiral, then you don't have any enantiomer. That is the like the reflection of that molecule will be superimposable on that object. But if you have if you have a chiral center, then the molecule will have enantiomers, that is d and l enantiomer for the case of amino acids. So now we will find out the different amino acids. Based on this r, different r, we will have 20 amino acids, and we will have name of the amino acid, then is three letter code and one letter code. That is the way we represent it in the terms of a three letters or one letter code. So if r is equal to h, then the name of the amino acid is glycine, which we represent as gly and then g. So here, if you see, is this molecule chiral?
No.
No, because we have two similar substituents. So glycine is a chiral amino acid, the only a chiral naturally occurring amino acid. Now the next is, if R is equal to CS3, then the name of amino acid is alanine, and the three letter code for this is ALA, and the one letter code is A. Now if R is equal to isopropyl group, then the name of the amino acid is valine, and the three letter code is VAL, well, and then one letter code is V. So we have a positional isomer of this valine amino acid, where, sorry, not of valine, uh, we have this amino acid called leucine, and it has an positional isomer. So their names are leucine and isoleucine, and these are the codes. Okay. So these are five amino acids. One is glycine, then second is alanine, third is valine, fourth is leucine, and fifth is isoleucine. So these five amino acids has their R group as an aliphatic group. Okay. Now the second category is where R group is aromatic in nature. So here, so similar to methyl, like in the alanine amino acid where we have CH3, one of the hydrogen atom is now being substituted with this phenyl ring. So now because of this, we call this amino acid phenylalanine. The three-letter code is PHE, and because it pronounced as F, so for F, so its one letter code is F. Now the second one is trypto tryptophan, which has a phenyl ring, and then an indole group. This is indole group, which is bound to CS2. So if you directly attach this moiety to a carbon atom, then you will have a steric hindrance. So nature has a solution for it. It always has a CS2 joining unit. Okay. So here we get, sorry, TRPN, and the code is W. Next is CS2. Then we have CS2 and a para substituted phenyl ring, where we have hydroxy group on the para position. So this is tyrosine amino acid. These are the three-letter and one-letter codes. Then the third group is polar functional groups. Structure, name, their three-letter code and one-letter code. First is CS2OH, and its name is threonine. Then spargine. So these are the functional groups, polar groups in these these amino acids. Now the fourth group is acidic group. First is CS2COOH, that is aspartic acid. Tannic acid, glutamic acid. Then we have basic groups. Basic groups, structure, name, three letter code, and one letter code. So first is CH2, CS2. This is arginine, CH2. Sorry, this is lysine. Then we have this CS2, CS2, NH, C double bond O, C double bond N. This is guanidine unit. This is called histidine. Then we have special groups. We have one more. I'm not using eraser here because there's some problem with that. We have CH2, this imidazole group. This is histidine. This is special group. first has this thiol group, cysteine, cis, and then this has C. We have one more sulfur based amino acid which is methionine, and we have one selenite based amino acid also which we call selenocysteine. And then we have a amino acid which is like uh, 3D molecule, that is it is out of plane. If in a if in any protein you want to introduce any turning, like rotation of that molecule, you introduce this amino acid there, and this is very important amino acid which we call proline, pro, and the name is P. So these are 20 amino acids. I hope you are taking a note of this. I might have made some mistakes in between. Sorry about that. So the next question is, no, no, the earlier questions were not discussed. So now can you answer the second question? Anita, are you there?
Yes, ma'am.
So can you answer the second question now? Of the 20 standard amino acids,
Staying on the methionine.
yes,
Option C.
no. Read the question carefully. Of the 20 standard amino acids, only dash is not optically active. Optically active means that the molecule which has an achiral center. So the only amino acid which is achiral is glycine. So glycine is the right answer. But now, what is the reason of that? Its side chain has two same substituent, which is hydrogen atom. So the right answer is B.
Okay, ma'am.
Okay. So the next question is, two amino acids of the standard 20 contain sulfur atoms. They are, so now only we discuss the 20 amino acids. Tell me which of them have sulfur atom.
Option C.
Methionine and cysteine. Very good. So the next question is, all of the amino acids that are found in proteins except for proline contain. So as we discussed, each amino acid has two functional groups which are very common. Which are those? That is carboxylic acid and amine group. But as we
Hmm.
discussed right now, that in the proline we have this moiety, and this is not primary amine. This is secondary amine. Okay. So here in the proline we don't have amino group. So the right answer is A. Now the next question is, which of the following statements about aromatic amino acids is correct? First is all are strongly hydrophilic. Second is histidine's ring structure results in its being categorized as aromatic or basic depending on pH. No, only based on pH we can define an amino acid as acidic or basic, but not aromatic. Now the third is, on a molar basis, tryptophan absorbs more UV light than tyrosine. So that doesn't define the aromaticity. Then D answer is, the major contribution to the characteristic absorption of light at 28 nanometers by protein is the phenylalanine R group. And the E is, the presence of a ring structure in its R group determines whether or not an amino acid is aromatic. So if a molecule has a aromatic ring, this E answer seems to be relevant. That is, we have a delocalized aromatic ring, then only we define that amino acid to be aromatic, not based on any pH. Okay, and whether the molecule absorbs in UV region or at a particular wavelength, that is not going to define a, a, a category of amino groups, that is aromatic, aliphatic, acidic, basic, etc. Is it clear?
Clear, ma'am.
Okay. So the next question is, which of the following statements about cysteine is correct? So cysteine forms when this CH2SHR, that is this is methionine group, sorry, cysteine group. So only if you see this CH2SH group will combine in the oxidized state, will form CH2SS bond. So we can say A is the right answer.
Ma'am,
Yes.
uh, it, it seems option E is also right.
Cysteine B,
Last option.
last option. Two cysteines are released when a CH2SS2 disulfide bridge is reduced to CH2SH. So if this bond breaks, then no, here we will get cysteine. Cysteine is forming cysteine. Okay, this is cysteine. So if you break this bridge, then we will get this cysteine amino acid. Okay, so here the name is incorrect.
Okay, ma'am.
Okay, so now tell me which structure is wrong for an L alpha amino acid. So here you have to find out the L enantiomer. So firstly, we will find it out for these two because they are already represented in the dash wedge form. So H is on the back. Now we have to assign the positions. CO minus will be 1. So here they are represented in their Jupiter ionic forms. because in the same molecule we have acidic COH group and basic amino group. So they exchange their hydrogen atoms. That is, basic group will abstract the hydrogen atom from the carboxylic group. So the now here CH3 will be second and NH3 plus will be 3. So here we are moving anti-clockwise. So this is L amino acid, L enantiomer. Similarly, here 1, 2, 3. So this is D enantiomer. So the right answer, which structure is wrong for an L alpha amino acid? This is clearly D. Okay. Now you can also present this as COO minus H CS3 NH3 plus. The representation is wrong. COO minus H NH3 plus. This is CS3. 1, 2, 3. So this is L enantiomer. COO minus H NH3 plus and CS2 SH, because these two are in plane and these are towards and away from the observer. So this will be second and this will be third. So this is also L enantiomer. So the right answer is D. Okay, is it clear?
Yes, ma'am.
Now the next question is the helical structure of protein is stabilized by. First is dipeptide bonds, hydrogen bonds, ether bonds, and peptide bonds. Do you have any idea? So these are the general questions I have taken. These are not directly relevant to the this course. So I have just taken the basic biological questions on these protein and amino acids. So can you answer this question?
Option A.
So the answer is hydrogen bonding. In the helical structure, so in the main lecture also, it has been cleared that these are the chemical bondings present present in the proteins, enzymes, etc., which stabilizes them. First is hydrogen bonding, second is salt bridge, third is hydrophobic bonds, pi-pi interaction, and fifth is weak interactions such as van der Waal interactions. So in helical structure, which is like this, we have proteins. So here you can see there are hydrogen bondings between these C double bond O carboxyl group and this amine group, that is NH. So because of the hydrogen bond between these oxygen of carbonyl group and hydrogen of amine group, the alpha helix structure of this protein is stabilized. Okay, so this is the hydrogen bonding which is present in the molecule which stabilizes it.
Ma'am, screen is not shared.
I've shared it. It is visible on my screen. You can't spot it.
Yes, ma'am.
Okay, see, this is the carbonyl group here, C double bond O, and this is the NH bond. So here, these hydrogen bonds are present, which stabilizes the molecule. This alpha helix structure. Is it clear?
Screen is not visible to me, ma'am.
Is it visible now?
Mm,
No.
no, ma'am.
Okay, okay, no problem. I'll add the image in the notes. Okay.